Hi, my name is Neil Frick. I'm the Senior Vice President of Medical Programs and Information at the National Bleeding Disorders Foundation. I would like to welcome you all to this recorded version of Gene Therapy, Real-World Experiences and Current Challenges. This was a session that took place live on August 18th, 2023, during the National Bleeding Disorders Foundation's recent annual conference in National Harbor, Maryland. I have the distinct pleasure of introducing Dr. Radoslav Kazmarek. Dr. Kazmarek is an assistant research professor of pediatrics at the Gene and Cell Therapy Laboratory of Wells Center for Pediatric Research at the Indiana University School of Medicine. Dr. Kazmarek chairs the Coagulation Products Safety, Supply, and Access Committee of the World Federation of Hemophilia and is a member of the ISTH SSC Working Group on Gene Therapy, as well as a member of the European Hemophilia Consortium's Medical and Scientific Advisory Group. Dr. Kazmarek's talk is entitled Gene Therapy for Hemophilia, Progress and Conundrums. Welcome, Dr. Kazmarek.
Hello, thank you, Neil, for this kind introduction. In this presentation, I'm going to give you an overview of the current status of gene therapy for hemophilia and several stumbling blocks that have kept it from becoming where we need it to be and what we can do about it. Before we take a deep dive into gene therapy, let's take a quick look at the hemophilia treatment landscape. We have a molecular revolution in full gear right now with several new drugs approved in recent years and more on the way. What drives this innovation? Well, it has been driven by the need for better protection from bleeding, ideally steady state level protection, which is something that the standard factor replacement therapy cannot achieve. The ongoing revolution started with the arrival of extended half-life concentrates, which facilitate prophylaxis, but are still limited by seesaw pharmacokinetics, by peaks and troughs. Then in the 2010s, a number of non-factor therapies entered clinical trials, one of which has been approved and widely adopted. Most of these new approaches offer easy subcutaneous injections and less frequent dosing. And for the first time, they break the paradigm of seesaw pharmacokinetics, of peaks and troughs. And most of these completely flip the script in the way we restore hemostasis. However, their vastly different mechanisms of action give rise to uncertainties about efficacy and safety. Also, they do not normalize hemostasis, and they cannot be measured using the legacy assays that have long been established in the factor replacement therapy. So the quest for better therapies and ultimately a cure, somewhere along the line, has continued. In the last decade, we have seen the first evidence that gene therapy for hemophilia A and B might be able to fix hemophilia for a long time. When several gene therapy programs that use adeno-associated viral vectors started making some headway, two of which have been approved in the last few months. But it was not an easy journey. There were many obstacles and failures along the way. The origins of gene therapy for hemophilia really go back to the cloning and expression of the factor 8 and factor 9 genes in cell lines, which opened the possibility of creating a cure for hemophilia that would deliver a correct version of the factor 8 or factor 9 gene to the patient's cells in their body, and those cells would then use the gene to make functional cloning factor. For the next couple of decades, the new field tried different viral vectors encoding factor 8 or factor 9 in primary cell lines and then in small and large animal models. Some of them reached the clinic, but they didn't work. The prototypical AAV serotype 2 was among those early viral vectors tested. And despite its failures, it seemed to be the safest and most promising one at the time. In preclinical studies, it produced lasting factor 9 expression upon intramuscular or intraportal injection, first in mice and then in dogs with hemophilia B. But these results did not replicate in the first two clinical trials. In the muscle trial, factor 9 levels were stable but low, and dose escalation was not feasible because it would require dozens upon dozens of intramuscular injections. Also, a lot of the factor 9 proteins stayed in the connective tissue between muscle fibers, excuse me. where it was likely bound to collagen 4. In the liver trial, circulating factor IX levels were therapeutic but transient. They rose to 10 to 12%, which was very impressive at the time, but then they dropped to the baseline within 10 weeks, and the decline was accompanied by transaminitis. It later turned out that the expression loss and transaminitis were caused by cytotoxic cellular immune responses against AAV capsid antigens presented by transduced hepatocytes, indicating that the problem could be solved with immunosuppression. Meanwhile, the choice of AAV serotypes that could be used for gene transfer expanded, and they worked well in correcting hemophilia in mice and dogs, but it took several years before AAV vectors for hemophilia were tested again in humans. When they finally returned to the clinic in 2010, it was a big comeback. In that trial, several individuals with hemophilia B received an AAV8 vector encoding factor IX. It targeted the liver, and like the previous liver trial, ALT started going up, and factor IX expression started dwindling down in some individuals, but it was rescued with a course of steroids. The last report on those individuals was 8 years after the therapy. They had stable factor IX levels, and anecdotally, they remain stable now more than 10 years later. That success informed and encouraged several other trials testing AAV vectors encoding factor IX or factor VIII, many of which are still ongoing. They all administer AAV vectors targeting the liver in a single bolus injection in a peripheral vein. These vectors are manufactured using human embryonic kidney or insect ovarian cell lines. The first hemophilia B studies used transgenes that encoded wild-type factor IX, and they saw factor IX increases within moderate and lower mild ranges. Better results came after incorporating the hyperactive Padua variant of factor IX, which has a single amino acid substitution and has since been used in all the leading hemophilia B trials. The change boosts factor IX activity about 8 times, producing higher factor IX levels without increasing the vector dose. So by using the same vector doses as those used in the normal factor IX trials, they raised the average factor IX levels to the higher mild and normal ranges. The same team that revived hemophilia B gene therapy also created several AAV vectors that encoded B-domain deleted factor VIII. In 2014, one of them was chosen for clinical development and became BMN 270. And this product was approved by the FDA this year. It was the first clinical trial that reached normal factor VIII levels in some individuals, and several other vectors showed similar potential soon after. 2 clinical programs for hemophilia A and 2 for hemophilia B have advanced to phase 3 clinical studies, and early phase studies have generated multi-year follow-up data. And now, after decades of experimentation and disappointments, after many fits and starts, the first gene therapies for hemophilia A and B have been approved in the last few months. But is this the end game of our pursuit of a cure for hemophilia? Well, not quite. Data from large cohort phase 3 studies have substantiated several concerns about variability, durability, predictability, and safety that arose at earlier stages. Many individuals, and in hemophilia A studies, most individuals had elevations of liver enzymes. Increased ALT is the most common adverse event, typically managed with corticosteroids, as in the original 2010 trial, and more recently also with other forms of immunosuppression. These elevations are not associated with any symptoms. They are mostly mild and short-lived, between 1 to 3 times above the upper limit of normal. But in some cases, they were as high as 10 to 20 times over the upper limit of normal, or stayed high for as long as over a year. Their incidence appears to be higher with higher vector doses, And in hemophilia B trials, some of these cases were linked to cytotoxic T cell response toward AAV capsid proteins, and they variably responded to immunosuppression. In hemophilia A studies, the correlation between the cellular immune responses and ALT elevation is rather weak, and immunosuppression has been less effective at managing them compared to hemophilia B studies. Only one hemophilia A study found evidence of cytotoxic T cell response, and yet in hemophilia A trials, liver enzyme elevations have been more common. The durability of efficacy is clearly different between factor VIII and factor IX studies. This is what really goes to show that hemophilia A gene therapy may be faced with additional challenges to those facing hemophilia B. In hemophilia B studies, factor IX expression has been remarkably durable. Participants of the original 2010 trial have maintained stable factor IX expression, and more recent trials using AAV constructs that encode factor IX PADWA have had similar success. In contrast, factor VIII expression has declined in almost all individuals who received their highest vector doses and continues to decline. In the longest running hemophilia A trial, 6 years after vector dosing, the median factor VIII levels have dropped 8.5 times. Individuals who received lower vector doses have had a more stable expression, but their factor VIII levels were markedly lower in the moderate to lower mild hemophilia range. In all trials, factor VIII and factor IX expression is highly variable, to the point that means and medians poorly represent levels achieved by individuals, from no response at all to moderate increase to levels across mild hemophilia and normal ranges, and far above the upper limit of normal. This is especially true for hemophilia A gene therapy. The causes of transaminitis, factor VIII expression decline, and wide variability remain obscure. We don't really understand much about these observations. Some expression variability is to be expected because gene therapy is not a simple process. It is not like making a protein drug outside the body in a controlled, fine-tuned environment of bioreactors. Here we are giving the body a new instruction to make the protein itself. And there may be a lot of variation in how it goes about doing it. For successful synthesis of transgenic protein, upon gene transfer, the vector has to reach the target cell, get inside it, travel to the nucleus, get inside it, and release its genetic payload, which has to undergo a complex reassembly to permit transcription and translation of the transgene product, which then has to go through multiple maturation steps before it leaves the cell to perform its function in the circulation. Now, how well each of these many steps goes may depend on individual sets of genetic and environmental factors. Some of these steps might be amenable to modulation with existing pharmaceuticals, which could partially reduce the variability of expression, but probably not eliminate it. Suffice to say that there is large variation of endogenously produced factor VIII levels, both within and between individuals with moderate or mild hemophilia A, carrying the same factor VIII mutations. At least 17 genes determine individual factor VIII levels. So this is not something we can solve easily. But we should try harder to solve it, at least partially, and reduce variability. Because none of those options have been explored. And despite the fact that the vast majority of individuals have shown reduced bleeding rates and have not needed the standard therapy, individual outcomes are unpredictable, whereas factor level is the key determinant of success of gene therapy. It would be great if you could give another dose to those individuals with suboptimal therapeutic responses, but we can't because of the universal development of high titer and cross-reactive anti-AAV antibodies, which can persist for at least 15 years. This precludes repeat dosing, even with other serotypes, because of cross-reactivity. Potential solutions such as plasma exchange or proteolytic antibody depletion are at early stages of development, and they will be difficult to integrate in routine practice. Conversely, patients on the other side of the variability spectrum are faced with the risk of phenotoxicity, insofar that supraphysiological factor levels may lead to thrombosis. High factor levels prompted a pause of one factor VIII gene therapy program and prophylactic anticoagulation in some individuals, while a participant of one hemophilia B program required both acute and prophylactic anticoagulation when his factor IX levels rose above 500%. So potential excessive factor levels is also something to bear in mind because once you dose, you can't turn it off. Since the discovery of cytotoxic T-cell mediated killing of transduced hepatocytes, we have learned much more about immune responses to AAV gene therapy. Most of it we know from basic research, but some of the findings have already manifested in the clinic, pointing to potential mitigants. Toll-like receptor 9 mediated sensing of a vector DNA cargo has emerged as a key mechanism triggering immunotoxicity and expression loss. TLR9 is an innate immune sensor of unmethylated CpG sequences in DNA, which are pathogen-associated molecular pattern. The most advanced clinical programs stripped their vectors of those elements, but the most recent unsuccessful trials inadvertently increased the number of CpG sequences in their vector constructs, leading to factor IX expression loss within 3 months from dosing that did not respond to immunosuppression. Complete removal of CpG sequences is difficult, and the remaining CpGs potentially contribute to residual immunogenicity of otherwise CpG-depleted constructs. Analysis of the discontinued BAX335 study suggested that interleukin-6 receptor is a promising target for mitigation of AAV immunotoxicity because the only participant who had sustained factor IX expression also had a missense mutation in the IL-6R gene. Non-clinical research has identified several other targets that may be fertile for clinical translation. One hypothesis links liver enzyme elevations to the very large number of vector particles that the transduced cells have to deal with. This is one of the reasons why using lower vector doses might help reduce toxicities. One way to lower the doses would be by improving transduction efficiency, and there is room for improvement. It is important to understand that AAV transduction is an extremely inefficient process. More than 99% of vector particles perish in the cellular garbage disposal pathways before establishing transgene expression in the nucleus. And recent outbreaks of wild-type AAV-mediated hepatitis in children indirectly point to the role of high dose. Because almost all severe hepatitis cases had high levels of AAV2 in the livers or blood, and co-infections with helper viruses triggered intense replication of AAV2 in the affected individuals, possibly reaching or exceeding numbers otherwise only seen in the context of gene therapy. The studies also identified an HLA2 haplotype that correlated with hepatitis. This suggests that HLA typing might have value in predicting the outcome of gene therapy. In hemophilia A gene therapy, all these problems are compounded by unique challenges of the factor VIII biosynthesis and factor VIII encoding AAV vectorology, which may contribute to liver enzyme elevations and the prolonged multi-year decline of factor VIII levels. Unlike factor IX, factor VIII is primarily produced by the liver sinusoidal endothelial cells, or LSECs, not by hepatocytes. And yet, AAV vectors encoding factor VIII target hepatocytes, and thus factor VIII transgene expression is ectopic. Factor VIII is a large and complex glycoprotein, notoriously difficult to express in heterologous cells, even compared to other similarly sized and structured proteins. Several bottlenecks have been identified behind the poor factor VIII expression. including transcription repressing elements in the factor VIII gene, tendency to misfold, aggregate, and degrade, the requirement for a specific receptor complex to traverse the cellular secretory pathway, and poor stability in the absence of von Willebrand factor. Importantly, misfolded factor VIII activates unfolded protein response, which can lead to cellular stress and death. New generations of episomal AAV gene therapy for hemophilia A address those imperfections by imperfections of factor VIII by using its engineered variants, which produce higher factor VIII levels, typically due to improved secretion, meaning that they could permit using lower vector doses. Porcine factor VIII has long been known for much better secretion than human factor VIII, which led to development of factor VIII ET3, a hybrid human-pig factor VIII variant, the sequence of which is 9% porcine. Clinical studies administering AAV or lentiviral vectors encoding ET3 are about to begin or have already started. Several amino acid substitutions have been shown to improve factor VIII secretion, and multiple preclinical studies in animal models are testing those molecules. Will they solve the problem of factor VIII decline? Nobody knows. Our vague idea about what happens doesn't go far beyond speculation and hypothesis generation. Recent basic research studies point to a complex interplay of cellular stress, the immune system, and protein biosynthesis elements leading to translational shutdown. Others suggest that epigenetic modifications of episomes mediate the factor VIII decline, and perhaps both studies are correct. For the time being, we still have more questions than answers. Liver biopsies will be critical to understanding AAV gene transfer biology in humans. Only one such study has been published so far. The results were encouraging, but this study didn't look sooner than 2.5 years after dosing. Future studies need to look at earlier time points, ideally in a time series to capture the peak signs of toxicity in the first year. One important feature of factor VIII is that it tolerates engineering within the B domain, which is not needed for its procoagulant activity, but makes up a large portion of the factor VIII protein. Removal of the B domain has improved expression and permitted inclusion in AAV transgene cassettes, producing 5 kilobase long constructs as compared to over 5 kilobase long full-length factor VIII. However, B-domain deleted factor VIII transgenes still exceed the optimal AAV capsid packaging limit. Vector packaging efficiency dramatically drops with transgene extension beyond 4.7 kilobases. So oversized vector preparations are mixtures consisting mostly of vectors harboring truncated payloads. Upon transduction, some of those fragmented vectors undergo repair in the nucleus and may be assembled into functional monomeric and concatemeric circular episomes by the cellular DNA processing machinery, which is essential for establishment of persistent transgene expression. However, these extra steps reduce the efficiency of an already inefficient process. So this necessitates higher vector doses. In a sense, factor VIII gene therapy is a compromise between the size of transgene cassette and vector dose necessary to induce meaningful transgene expression. The long-term safety of AAV gene therapy for hemophilia has been encouraging so far, both in humans and large animal models, but it needs careful attention. AAV have long been misperceived as non-integrating vectors due to the formation of episomes and the relatively low rate of chromosomal integration, estimated between 0.1 and 1%. However, in absolute terms, this integration may still result in millions of events in the liver, with vector doses ranging from trillions to quadrillions of particles, raising questions about genotoxicity and oncogenicity risks. There have been 4 cases of cancer in AAV clinical trials for hemophilia to date. None of them were found to be related to AAV vector integration, but comprehensive genetic analysis did find integration events at rates that were close to previously predicted. Importantly, despite their liver tropism and liver-specific expression, AAV vectors may transduce other tissues, so monitoring should be geared toward detecting other potential toxicities. Although the evidence of malignancy risk is limited to neonatal or liver-damaged rodents, it is an unknown that calls for vigilance and long-term, preferably lifelong monitoring of recipients. So overall, there is a lot we still don't know. The advance of gene therapies toward commercialization has far outpaced our understanding of AAV gene transfer biology in humans. The mechanisms of wide variability, waning efficacy in the case of hemophilia A, and liver toxicity are largely mysteries. There are plausible hypotheses, but we haven't really put our finger on what's happening. Neither in preclinical nor in clinical studies, in which we barely scratch the surface. We need to understand these problems better mechanistically to develop effective mitigation strategies to ensure safety and the best possible outcomes. Minimizing AAV wastage throughout its journey to the nucleus and improving factor expression could lower vector doses by orders of magnitude, and that in turn could reduce toxicities. The dose is likely critical because toxicities grow in frequency and severity with increased vector dosing. And although liver toxicities in hemophilia trials have been mild and transient, we shouldn't be too relaxed about them until we understand the mechanisms. The threat of chronic hepatitis C had long been underestimated for similar reasons until it wasn't, when it turned out deadly years later. So as long as we don't know something, we should acknowledge we don't know it, and we should try to understand it rather than ignore it, lest we miss something pernicious going on. The field will also benefit from more transparency and data sharing, as well as timely publication of animal and clinical data, including negative results. The discontinued BAX 335 program demonstrated that unsuccessful studies may offer important learnings. A framework of known unknowns has been proposed to organize thinking around these concepts. It's a very helpful way of thinking about their short and long-term safety. We have published several communications highlighting these matters over the last few years, in which we encourage the field to abandon Panglossian or overly optimistic thinking in developing of AAV gene therapies, and to look at them a little more critically. Some limitations of the episomal AAV gene therapy are inherent in the technology, and may need to use alternative gene transfer systems to be overcome. Several alternatives are being tested, mostly in preclinical studies. Some still use AAV or a combination of viral and non-viral vectors in the form of lipid nanoparticles. Some combine genome integration with transgene delivery by AAV or use a different viral vector altogether. And some are fully non-viral. Lentiviral gene therapy studies have already dosed first patients. They collect hematopoietic stem cells from participants, transduce them with lentiviral vectors encoding factor VIII, and infuse them back to the patients, where they go on to home into bone marrow and generate megakaryocytes or monocytes expressing factor VIII. They promise durability, and additionally, in preclinical studies, the platelet approach worked in animals with factor VIII inhibitors, but they involve aggressive conditioning to make room for transduced stem cells, and that aspect needs work before. When you compare it to a simple intravenous injection of AAV vector in a vein. There are dual AAV or AAV and lipid nanoparticle approaches in development that consist of an AAV vector encoding factor VIII or factor IX, and another AAV or lipid nanoparticle delivering CRISPR-Cas9 machinery for targeted transgene integration in the genome. Most importantly, these approaches overcome the transgene dilution effect, which has been a barrier for episomal AAV gene therapy in children. who have the most to gain from gene therapy. But these methods still, at least partially, depend on AAV vectors, so they will be faced with some of the same problems that have hindered episomal AAV gene therapies. And then there are fully non-viral approaches, which use lipid nanoparticles to deliver a transposase and a factor VIII encoding transposon in a dual LNP system. Or there is a technology that uses just a single lipid nanoparticle to deliver close-ended DNA encoding factor VIII. These closed-ended DNA, or sedna, resemble AAV transgene cassettes, but are packaged in lipid coating instead of viral capsids. These approaches may solve the problem of unpredictability and variability, and also eligibility, because there is no pre-existing or post-infusion neutralizing immunity against LNPs. So these vectors can be redosed and titrated to achieve desired transgene expression. The downside of transposon approach is random integration in the genome. There is preference for intergenic regions, but nonetheless, this isn't targeted gene insertion as in gene editing. Also, LNPs unload their payloads in the cytoplasm before reaching the nucleus, unlike AAVs. So this must impact transduction efficiency, because transgenes need to reach the nucleus to establish expression. sedna LNPs looked impressive in mice, but disappointing in larger animal models. One gene editing approach has been tested in the clinic already in a single individual with hemophilia B. The study administered 3 AAV vectors, 2 encoding zinc finger nucleases and 1 encoding factor IX. But that individual never achieved detectable factor IX. So all things considered, each of these alternative systems solves one or more problems limiting episomal AAV gene therapy, but none of them solves all of those problems. And each comes with their own limitations. So these technologies have yet to mature and prove themselves really as viable alternatives for the current wave of AAV gene therapies. So where does this leave us? We might have a cure for some individuals with hemophilia B, at least for those who will achieve curative even-keeled factor IX levels. Those who did not lose expression in the first year after dosing have maintained it for years and decades. For hemophilia A, the cure appears temporary, and the therapeutic effect might eventually dwindle to a trickle, creating new challenges and requiring return to prophylaxis. Several subgroups in the community are missing out on the opportunity altogether, including children, people living with HIV, patients with current or past inhibitors, and those living in places where cost will be prohibitive. Future efforts should aspire to include the long underserved and underprivileged groups. AAV gene therapy has proven to be a powerful platform for clinical gene transfer, but it is still a work in progress. With much room for improvement. AAV transduction and transgene expression engage cellular machineries in ways that are poorly understood and need a lot more preclinical and clinical research. We also need more transparency and timely publication of data. Good judgment comes from experience, which comes from poor judgment. And I believe that collectively we are past the poor judgment stage, and we have quite a bit of experience, but that experience is useless if kept secret. We need more data sharing to exercise good judgment moving forward. For gene therapy to really work as a cure, it needs to be able to completely close the gap between how much hemostatic correction a therapy can provide and how much is needed to live a life independent from treatment. That should really be our touchstone of a successful gene therapy, whether it can reliably turn an individual with hemophilia into somebody without any need for additional hemostatic treatment whatsoever, even when they experience trauma or need surgery, because the goal is a cure, not another treatment. Thank you for your attention.
On behalf of the National Bleeding Disorders Foundation, I want to thank you, Dr. Kazmarek, for an excellent presentation. And thank you for being a part of our medical track at the 2023 Bleeding Disorders Conference. And thank you for joining us.
It was a pleasure. Thank you.